We know that the DNA duplex is 20 angstrom in diameter. The duplex is coiled plectronemically in a right-handed manner. I told you that it looks like a rope stair in twisted form so as to form a spiral, and this coiling produces alternate major and minor grooves. The major groove is 22 angstrom long and the minor one 12 angstrom long. Each turn of spiral has a distance of 34 angstrom. Each turn means starting from a point to 360-degree turn. When completed, it means a complete turn. Each turn of the spiral has a distance of 34 angstrom, and within this length, there are 10 deoxyribonucleotides in each chain. That means the average distance between adjacent deoxyribonucleotides is 3.4 angstrom. And the pairing is formed in the DNA double helix between the purine of one strand and pyrimidine of the second strand in such a manner that the width of the DNA molecule remains 20 angstrom all along its length. If it happened that a double-ringed purine formed a hydrogen bond with a double-ringed purine, then that place would be wider along the DNA length. Such places where purine-purine bonds are supposed to form would be wider, but where pyrimidine bonds would be formed, or are supposed to form, would be thinner. That means along the length of the DNA, the 20 angstrom diameter would not be maintained, but actually there is 20 angstrom diameter throughout the length. That means one purine combines with one pyrimidine, and that happens in such a way in all DNAs, regardless of the species. Whichever species the DNA may belong to, the number of adenine residues is equal to the number of thymine residues. That means A equal to T in any given species, and the number of guanine is equal to the number of cytosine residues. That means G equal to C. This relationship proves that the sum of the purine residues, means A plus G, is equal to pyrimidine residues, means T plus C. This quantitative relationship is called Chargaff's rules, because Chargaff found out this relationship, and while proposing the model of the DNA, Watson and Crick took the help of this Chargaff's rules. Not only Watson and Crick, Rosalind, Franklin, etc., all those took these rules help and these rules were confirmed by many subsequent researchers. And I told you that adenine combines with thymine and guanine with cytosine. AT bond is double hydrogen bond. What is a hydrogen bond? I will try to give you a small video to make you understand how hydrogen bonds are formed. However, some still pictures will also be presented to you for your better understanding. And now let us know that the double hydrogen bond formed between adenine and thymine, and the triple hydrogen bond is formed between guanine and cytosine. And when a DNA molecule is heated to a temperature of 50 degrees Celsius, the two strands get separated because of the breaking of the double hydrogen or triple hydrogen bonds with the at the cost of the heat energy applied. And this process is called denaturation, because in natural condition the two strands remain together. And when the heat is removed again, once again when the two strands get the normal temperature, they have a tendency to reunite to form the DNA strand once again, and this is called renaturation. So DNA has this property also. And as for the nucleotides, the DNA has a polarity. Polarity means all the phosphodiester linkages have the same orientation along the chain, giving each linear nucleic acid strand a specific polarity, a distinct 5' 3' ends. The 5' end lacks a nucleotide, it ends there. On the 5' carbon, a phosphate forms a phosphodiester bond, and that is the end point of a DNA. No more nucleotide is there at the 5' end, and at the 3' end also there is a hydroxyl group and no more nucleotide is there. So the 5' end lacks a nucleotide and the 3' end also lacks a nucleotide at the 3' position. And the backbone, sugar phosphate backbone, is covalently formed, and as the phosphoric acid moieties are ionized and the hydroxyls are ionized, this backbone is hydrophilic, and as the phosphoric acid moieties are ionized, this DNA molecule is called an acid, hence it is a nucleic acid. And in the inner side, the purines and pyrimidines form hydrogen bonds to remain stacked together one after another like the rungs of a ladder at a distance of 3.4 angstrom, I told you. And this stacking force enable them to remain stable in that form, and that inner side of the DNA, that means the sugar, the two sugar phosphate backbones at the two sides, and in the inner side there are the one pyrimidine with a complementary purine, or one purine with a complementary pyrimidine. This inner side, this core part, is hydrophobic. That means the inner side of the DNA molecule is hydrophobic and the outer side is hydrophilic. This is also a property of DNA. And now I will give you some pictures to so that you can understand the structure of the DNA better. And then along with this audio and the previous one, I will give a small video also, so that you can understand some more points which cannot be explained in the audio. Thank you.